Who can introduce Dr. Pilla?
Yes, I will introducing Dr. Raffaele Pilla. Just
please. Please.
I would like to introduce Dr. Raffaele Pilla, San Joseph God Hospital, Italy, and his title will be Therapeutic Ketosis and the broad field of application for the ketogenic diet, ketonist application, and clinical update. And I would like to introduce his biography. Raffaele Pilla, pharmacy, PhD doctor, European, receives his master's degree in pharmacy at GD Anizuya University in Citizen Pescara, Italy in 2005, where he also served internships at the Cell Physiology Laboratory and Molecular Biology Laboratory. Thank you.
Okay. Thank you. Go ahead, doctor.
Thank you. Thank you very much for inviting me to this great webinar. I thank you for being able to share my experience and research I've been doing through these years. And I hope this is going to be of interest for everybody. I guess you can see my slides, can you?
Yes.
Perfect, perfect. Okay. So today I'm going to share my research experience. As now I am a pharmacist, I'm Italian, I work in Italy, but I've been doing a lot of research abroad. I've been a few years in France, in England, and in the United States. And I did my postdoc in the United States. This is where my story starts from. I say my story because it's actually a path I would like to drive you through. I've been working since 2010 with the Office of Naval Research. It's that branch of the Navy, of the US Navy, that is responsible for research. What do these guys do? They take care of technical missions, tactical missions, defense, attack, and rescue from submarines, as you can see from these pictures here. But specifically and notably, the ONR, or Office of Naval Research, is responsible for any sort of research within the Navy. We used to have a very special side of our lab, which was called the gas palace. The gas palace used to contain a lot of gas tanks, thanks to which we were able to reproduce, to mimic some very deep Navy SEAL dives, like in extreme conditions. Now, I don't know how many of you today at the webinar are familiar with the physiology of diving. How many? I don't know if there's any of you who is a diver, but I am a diver, and I can guarantee it's a very fun activity. But it can actually hide a lot of risks, life-threatening risks. In fact, if we do not surface properly, like very slowly, we can have a lot of problems, starting from the bends. You might have heard that, bends are accumulation of nitrogen in the joints or other sides of our body. And according to where it accumulates, it can generate, a disease or just a bothering in best case scenario. So as now we're breathing 21% oxygen and 79% nitrogen. We are just approximating. When we dive, these gases are under compression, and our body is only able to use the oxygen. Nitrogen actually gets accumulated into the tissues, and if we do not let it out properly, so if we do not surface in a very slow way, if we don't do any safety stops, we have issues because it gets accumulated into the joints. It can cause a lot of pain, or it can get accumulated into the optic nerve and can make us blind, or our spine, and can make us paralyzed, and can cause a lot of problems. So in order to avoid this problem, back in the 70s, the Navy started to introduce more oxygen into the tanks, so kicking off the nitrogen. This was able to, this actually allowed the Navy SEALs to stay longer into the bottom for their missions and to surface very fast, so in case of an emergency. But that caused a lot of reactive oxygen species production, and that resulted in many cases in what is called the central nervous system oxygen toxicity, which is basically some sort of convulsion of seizures, like in grand mal seizure in epilepsy, that can occur actually in two scenarios. Scenario number one is during hyperbaric chamber the hyperbaric chamber therapy, as you can see in this picture, or during a deep dive, for the military, for the Navy. Now, for those who are curious, I can show you this video. The guy on the left is with impure oxygen. He's going to have seizure. He's going to communicate to the doctor on the right that he's feeling dizzy. So he's taking off his mask and he's going to have seizures. He's at a depth of about 40 meters. Of course, this is just an exercise. It's not real water. It's a hyperbaric chamber. So as you can see, this guy is losing his consciousness. He's having seizures due to a hyperbaric oxygen at high pressure. If you look at his legs right now, he's having a tonic-clonic movement typical of grand mal epilepsy. See, right now, he lost his consciousness. He's not going to remember a single thing, and he's seizing right now. Imagine if this happens under the water. Well, if this happens under the water, that means pretty much death. I mean, it's pretty sure. This is why the Navy wanted us to study this phenomenon, wanted us to understand the physiology behind that, and wanted us to understand if there was any way to prevent that to happen. So when I moved to the States, the lab group that I joined was very expert into the research of solitary complex neurons that are responsible for respiration. When I got there, they decided to translate this research onto whole animal research. And this is why we implanted into the Sprague Dolly rats some radiotelemetry models that you can see on the top right picture, that were able to communicate to a receiver data on electroencephalogram, electrocardiogram, and electromyogram, plus physical activity and body temperature for every animal before, during, and after a dive. So we wanted to understand if there was any physiological marker able to tell us that something was going to happen in these animals before the onset of seizures. Just to show you what it looks like when a rat has seizures, I'm going to show you this video. It's the video on the right panel. This rat is going to have seizures within like few seconds. This is what it looks like. It's going to have a forelimb and head tonic-clonic movements. See, this is a seizure for a rat. He's unconscious right now. So very briefly, what we found was that 3 to 5 minutes before the onset of seizures, there was an increase in the tidal volume, which is the volume of gas a rat would breathe, and the respiratory frequency, which is the times per minute a rat would breathe. Both these parameters went considerably up 3 to 5 minutes before the onset of seizure. So the first, element that we published was that physiologically the respiration would increase. But now we wanted to understand how to prevent seizures to happen, because as I said, it was pretty likely that divers in these conditions would die under the water. So we started off to understand one of the ways that could be possible to prevent seizures. Antioxidants, they were very mild. I mean, for this type of seizures, probably this accumulation of reactive oxygen species, that would have been insufficient. So we moved on to anti-epileptic drugs, but there were so many side effects implicated into that. So we decided to move. That was not an option. So then we moved to the starvation ketosis, basically fasting, because fasting brings us into a ketogenic, metabolism, into ketosis, which is basically consuming not carbohydrates anymore, but fats or ketone bodies. And at the same time, we know that we can achieve ketosis in two main ways. Way number one, if we fast, if we do not eat, just drinking water. Way number two, through a ketogenic diet. Ketogenic diet is pretty much a diet from which we take off carbohydrates, or keep it very, very low amount, increase the fat up to 75%, 80%, and the rest, 20%, 15, 20% is about protein. Well, we could not give our Navy SEALs an ideal ketogenic diet because they were under stress during the mission. It would have been very, very hard to take the comfort food out of their diets. So we decided to synthesize in our lab a non-ionic precursor of ketone bodies called the ketone ester that was able to increase the ketosis level as well as a ketogenic diet followed for 2 or 3 weeks, but only in 30 minutes and no diet to follow. So the same level of ketosis just with one compound. We synthesized this in our lab and we decided to run the experiment. But before we go into that, I wanted to specify that the ketogenic diet is made of, as I said, about 75 to 80% fat, about 20% protein, and up to 5% carbohydrate if allowed. What should we eat during ketogenic diet? Pretty much what you can see in this slide. So any sort of fish, any sort of meat, any sort of nuts, butter, olive oil, bacon, eggs, and this kind of food, avocado, any green leaf vegetable that would be allowed. What does it cause? Pretty much a decrease in glucose and therefore in insulin. So a decrease in inflammation. There's so many benefits in being in ketosis, and if you haven't tried before, you probably wouldn't get to the point of understanding how beneficial that can be. But what would be the best level of ketosis, the optimal level? According to many studies, but specifically from one of the very first papers discussing this point, from Jeff Volek and this group, the optimal level of ketosis is in between 2 and 5 millimolars. Below 2, that would be kind of not enough or not sustainable. Over 5, that would reach the point of 8 millimolar, which would be an alert, because passing that point, we would be going on to ketoacidosis, which is a pathological condition. We wouldn't want to achieve that. But at the same time, if we have a functioning liver and kidneys, that would never happen. What are the benefits of being in ketosis? There's so many, but just to synthesize what are the best ones. First of all, a decreased central fatigue, but also an increase in the metabolism of our brain. That would be fabulous. I tried that several times, and for instance, I am able to concentrate much longer and much tighter. There is a considerable decrease in reactive oxygen species. Therefore, we could actually see a neuroprotective effect against epilepsy of ketosis. This is one of the most beneficial effects. But also an increase of biogenesis in mitochondria, and also an increase in insulin sensitivity, and also less lactate in muscles when we exercise. And there's so much to say about that. There's so many benefits. I mean, you can look them up on PubMed. I mean, there's so much that has been published on that. You might think that this is a new concept. This is actually not a new concept. It has been published, from a journal back in 1922 for the first time from the Los Angeles Times, speaking of the Johns Hopkins Hospital in Baltimore, for the children's hospital, because children that were refractory to medications for epilepsy were put on a ketogenic diet, so in ketosis. And in this way, most of them were able to completely get off seizures completely. In some of the cases, only partially, and in just a very little portion of cases, the ketogenic diet had no effect. But this is not even the very first report. The very first reports are back from 400 before Christ, from Hippocrates. In the Hippocrates corpus, he described that the people possessed by the demon, by the devil, those people were probably most likely, epileptic patients, but back in the days there was no diagnosis for epilepsy. These patients were put in isolation for a few days, so no food, just water. And after a few days in isolation, these people were not possessed anymore. So this is probably one of the very first reports on what ketosis and how ketosis can be beneficial and neuroprotective against this kind of neurodegenerative disease. Anyhow, if you have any interest into this topic, I would strongly suggest you to go read this comprehensive review from George Cahill. This review dates from 2006 and comprehends pretty much all information you can find on the topic. So back to our experiments. After we gave intragastrically this compound, the ketone ester, to our animals, we were able to see an increase of resistance against these seizures in our animals, in our rats, of about 600%. So can you imagine A rat that would seize after 10 minutes in pure oxygen, after the compound would seize 60 minutes later. So from 10 to 60, from 20 minutes to 2 hours. That was huge increase. So we published this paper in 2013, pretty much telling Navy that one of the possible ways to, to prevent seizures to happen would have been to give them this compound. So this compound would spike up the ketosis level and neuroprotect them against epilepsy. We also found that among the 3 ketone bodies that are very well known in literature, like beta-hydroxybutyrate, acetoacetate, and acetone, the most responsible for the anti-epileptic effect was the acetone. Then I moved back to Italy in 2013 and started working with Dr. Vigiano and Dr. Coppola, one of the most high, I would say the most expert in Italy on the topic of ketosis. And we've been testing the same compound that we had patented in the United States called the ketone ester onto a very well-known model of epilepsy for research called the pentylenetrazole on rats. And we found that there was a considerable increase in resistance against epilepsy in these models too. On top of that, we published 2 papers speaking of, that discussed about how ketone ester was able to increase the latency time to seizure for these animals. But let's go back to the literature very quickly. So many benefits in following a ketogenic diet for a number of pathologies, and this has been published over the years. I would say, first of all, the weight loss. In fact, if you want to lose a lot of weight, the ketogenic diet is probably, together with the Atkins diet, is probably one of the best ways to act on the metabolism. There's so much in the literature you can find. Then we move through other neurodegenerative pathologies like Alzheimer's disease, Parkinson disease, and even Lou Gehrig disease, which is amyotrophic lateral sclerosis. There's a lot of literature on that. So there is a high neuroprotective effect of ketones in these pathologies. Also, traumatic brain injury is implied in these therapies. But what is even most important is that it can be applied in oncology. In fact, glioblastoma multiforme, one of the most aggressive brain tumors, can be fought with the ketogenic diet, with ketosis, through a very specific mechanism. Actually, the mitochondria in the cancer cells have some defects. They cannot metabolize ketone bodies. Therefore, they will only use glucose. But if we deprive our organism of most of glucose because we take it off for the diet, and we force the healthy cells to use the ketone bodies as fuel, we basically cut off cancer cells from getting fed out of our sugar, because that wouldn't be enough. So only selectively healthy cells can be fed. So moving on, and there's lots of results on this topic. Now, speaking of epilepsy, there's so many results. I don't have time to discuss about that today, but you can find them a little in the literature, and that's so much to say and so many witnesses that can discuss about that. So when I moved back to Italy, I needed something to convince people, to convince my patients, as I'm a pharmacist, to follow a ketogenic diet, so increase their compliance to the diet. How do we do that if they're very attached to pasta, pizza, desserts, fruit, and this sort of food? Then I needed someone that could produce some, food items that would look like, their sugar-based food, or their dessert, their pasta, pizza, but they wouldn't contain that amount of sugar. And I found this in one company that was able to produce these compounds. And this way, I was able to increase the patient's compliance. Plus, I published a number of videos on YouTube speaking of the benefits of ketosis for those who are interested, of course, because most of people are not actually interested in following a ketogenic diet. And I published a number of short reviews speaking of the benefits of therapeutic ketosis. Plus, we obtained with the Ministry of Health the possibility to distribute this food for patients for free, so they don't have to pay, because the public health system will pay for it. So a very brief communication about some patients affected by cancer that we've been treating with a ketogenic diet with success. This guy, Marco, he had prostate cancer with rib metastases. He was put on a mild ketogenic diet, and only 2 months after he began this ketogenic diet, he had his PSA going from 1.62 to below 1. He's been for over 2 years on a ketogenic diet, and he had no cancer recurrence, no evidence of,
Okay, you
cancer
have 5 minutes. 5
Yes,
minutes.
thank you very much. Thank you. Okay, just because I have 5 minutes, these are all cases of people that, we've been treating of cancer, several cancers that have been on a ketogenic diet and successfully went through it and are under observation. I'm sorry I go faster, but otherwise I cannot get to the end. Plus, we've been, holding a lot of courses for registered dietitians to use the ketogenic diet in Italy for therapy. And the very last point for this presentation is the Parkinson disease. I've been researching a lot, and there is not much because and there's not too many neurologists that want to take the ketogenic diet into consideration for Parkinson disease, because one of the one of the side effects of ketogenic diet is constipation. Constipation can be pretty bad for Parkinson's patients. But anyhow, we understood there was not too much literature on that, and we understood that ketosis might have had a very good, neuroprotective effect for these patients. So we started a study with this group of people in the clinic very close to my house, actually, is in southern Italy. And we had stage 1 patients for Parkinson's disease. We have been analyzing their postural hand tremor and rigidity, leg agility, their speaking abilities, and so forth, before, during, and after a ketogenic diet. We wanted to keep them on a ketogenic diet for 3 months. Unfortunately, this data have only been started because these experiments, because then COVID came in and we had to stop everything. But so far, what we have found is that regarding their autonomic dysfunction, sleep troubles, and neuropsychiatric disturbances, there were a lot of improvements. Specifically, we've been using these tests they're very familiar for those who work with Parkinson's disease, and with memory and phonemic fluency and semantic fluency. I'm not gonna stop on the videos here. But then we found that according to this study published in New Zealand, we had very similar results. In this study, they put 38 patients on a ketogenic diet for 2 months and found that there was a decrease in urinary problems, pain, fatigue, daytime sleepiness, and cognitive impairment. But basically, we found the same thing. But most of everything, we found that ketogenic diet was safe to be followed for a longer period of time. So, long story short, I want to thank all the associations that have been helping me, helping the patients. And our next step with the Parkinson patients would be to use the ketogenic compound, the ketone ester, to neuroprotect against this neurodegenerative disease, for Parkinson's patients in the future. I want to thank the GLUT1 Association for their help, rareconnect.org for their help, and Dr. Cusano here, who has been helping me following a number of patients with the ketogenic diet. And very last thing, I want to present you 4 movies that might help us understand how bad sugar is for us. This is the first one, Fed Up, from the United States. This one is the second one, That Sugar film from Australia. This is another movie, very interesting, The Magic Pill, still from the United States. And this one is a homemade movie entitled Butter Makes My Pants Fall Off, which makes us understand how just eating butter and high-fat food can make us very thin and improve our blood work in general. So again, thank you to my European colleagues, American colleagues, and other colleagues. Thank you very much for your attention. If you have any questions, I'd be happy to answer.
Okay, thank you very much. Have you questions? I have one question. It's well known that epileptic discharge, the main reason is endogenic dysfunctioning calcium homeostasis. which bring negative resistance membrane permeability because membrane fluidity increasing, and all the factors which producing abnormal discharge is leading to increase intracellular cyclic IMP, producing sodium calcium exchange reverse mode, and increase lipid fluidity. So when neuronal swelling, then membrane becomes have a negative resistance. It's bursting, they having bursting activity. So you present very, very interesting data. I think that it really will be very useful if you will start from biophysical point of this study. For example, the metabolic regulation bursting activity of the cells, it's not synaptical, it's endogenic activity. And I recommend you even take my work in 1991, bursting metabolic regulation, bursting activity that Howard Wachtel from Duke University, they repeat this and they found out that really this function, sodium calcium exchange, leading to generation this effect. If you will apply any factor, for example, static magnetic field and other fields which stimulate cyclic GMP, it will be anti-epileptic effect. So this biochemical data, when you will analyze with all this data, because you know epilepsy is a rather old problem, and
Yes.
it was started on molluscal neurons, which has bursting activity, endogenic bursting activity, and the interburst of this is due to activation sodium potassium pump. Thank you very much.
Thank you. Thank you very much.
It was very excellent
Thank you.
lecture. There is any questions? If no, let's we will ask